Hello everyone, my name is Wenjun Li from Dr. Bingbing Chen's lab at Shanghai Tech University. The work I'm going to present is predicting potential damage occurrence after focused ultrasound-induced blood-brain barrier opening in rodents with machine learning. Acoustic feedback controlled focused ultrasound combined with microbubbles achieves a more reliable blood-brain barrier opening in rodents. As shown in this picture, in the feedback controlled FUS system we developed, a hydrophone is used to monitor acoustic emissions from stimulated bubble agents. Based on the detected acoustic emissions and our feedback control algorithm, the ultrasound power is adjusted automatically in real time. Current methods for assessing brain tissues damage occurrence, such as hemorrhage or red blood cells extravasation, include T2-weighted MRI and histology. They are all feasible, but the former requires the access of MRI scanner, and the latter requires complicated tissues harvesting and processing, which is time-consuming and not feasible for in vivo assessment. So our research question is how to achieve real-time assessing brain tissues damage occurrence after FUS procedure. In recent years, lots of fields use machine learning for prediction, classification, regression, and so on. Also, machine learning is used in biomedical engineering. For example, anomaly detection in renal image and an auto-label localization of fetal brain biometry plans in ultrasound. So our hypothesis is, can we use machine learning to predict brain tissue damage occurrence in real time after the FUS treatment? For using machine learning to predict tissue damage occurrence, the data is important. Here is our experiment setup. Our FUS system includes two parts, a FUS transducer driving subsystem and a hydrophone-based acoustic feedback subsystem. When we start sonication, bubble acoustic emission was received by the hydrophone in the center of the transducer. We process the data and feed it into our controlling algorithm. Based on that, the ultrasound power was adjusted in real time. This is our treatment protocol. At first, we require 5 seconds for data for the baseline, and then we start bubble infusion. In this project, we use 3 bubble agents. One is custom-made nanobubble with 280 nanometer, and another 2 are commercially available microbubble, Definity and Opticism. We started the FUS exposure with a low pressure and gradually increased it until reached the ideal level. When utilizing our real-time feedback control system, we keep the bubble emission at the desired level for 100 seconds. The ultrasound parameter was set 10 milliseconds pulse length and 1 Hz repetition rate. All the experiments were done at UT Southwestern Medical Center in the States. Here is the bubble data distribution map. We got 33 data sets from nanobubbles, 5 from Definity and 30 from Opticism. The blue color means health BBB opening, and orange means hemorrhage occurrence. Here is the representative frequency spectrum of acoustic signal for one treatment. It transforms by fast Fourier transform from our 100 acoustic data we received, and totally there are 20,000 data in 2 MHz frequency range. In the next step, just I told, we use the spectrum data comes from acoustic data to evaluate the importance of features, which is using a feature selection based on gaining importance of random forest. Then the selected data combined as a future dataset, plot all the damage not occurrence future data into the support vector data distribution model to detect damage case. The algorithm was validated with the leave one out method. The validation set is combined leave one not hemorrhage data out and combined and with the residual hemorrhage data. Since there are multi-features and small samples will make SVDD model not achieve good results. So we must to find a way to reduce the features at first. That's the problem is downsampling in the whole frequency range better than downsampling in the frequency range specifically corresponding to bubble behaviors. Based on this, we put optimism data into our algorithm at 0 to 2 MHz 80 times downsampling and 0.7 to 0.8 MHz 4 times downsampling respectively for verification. The latter is the range specifically corresponding to bubble behaviors. The results are listed in the table. We can find that the prediction of downsampling in the whole frequency range is not much different from that in its specifically corresponding to bubble behaviors. Also from the rock curve below, it can be seen that the AUC in the whole range is 0.79 and the AUC in the range specifically corresponding to bubble behaviors is 0.84. So there is no significant difference between two methods. However, 0.7 to 0.8 MHz 4 times showed a slightly better performance. Then we got another question. Can the machine learning trained from one bubble type can be applied across different bubble formulations? Now we first choose the key characteristics from the optimism data before training the SVDD model. Next, test the SVDD model by these future data, and then test the chain SVDD model by predicting tissues hemorrhage and analyzing the results using the same features on the deformity and the non-bubble. Here is the findings in this table. Based on the F1 score we obtained, we discovered that while the SVDD model trained using optimism as a training set, it can discriminate the hemorrhage data in optimism while it performs poorly on deformity and non-bubble. So we got that the SVDD model trained from one bubble type cannot be applied across different bubble formulations. Yes, it's our conclusions. First, machine learning based fast brain tissues damage occurrence with the SVDD model is feasible. And next, there is no significant difference between two data pre-processing methods, while the 0.7 to 0.8 MHz showed better performance, and the SVDD model trained from one bubble type cannot be applied across different bubble formulations. There are more works we need to do in the future. We should explore other methods and algorithms for different bubble formulations. And finally, I'd like to thank Chen Bing and Rajiv Chopra from UT Southwestern Medical Center and Qian Wang, Bingbing Chen from Shanghai Tech University for their support and guidance. Here is our Chen family. Thank you for your attention.